Hello, welcome to Ash Jottings. In this video session, we'll talk about factors which helps to stabilize the DNA structure. So let's get started. Factors that determine the structure of DNA. So there are a couple of factors like hydrogen bonding, hydrophobic interaction, hydrophobic interaction. Then you have base stacking, sorry, base stacking, K-K-I-N-G. Base stacking, then you have ionic strength of a solution on the structure of the DNA. So let's actually talk about each one of this. Before I head, I would like to um, explain you about what is denaturation. I hope and I'm sure most of you will be familiar with this term. What is denaturation? So the transition from a, so you know that the DNA is a double-stranded helix. Okay, it's very bad diagram. So when you actually pass on the heat or slowly start to heat the DNA solution which contains DNA, it starts to unwind. Okay, it's a very bad diagram. So it starts to unwind. Okay, and finally it will be two strands, right? So this is the native form and this is the disrupted form, or you can say denatured, disrupted, or denatured state. So state from a native to denatured state, the process from the native to the DNA transition from native to denatured state is called as denaturation. So as the denaturation process proceeds, changes in the property of the molecule occurs. For example, if you say, example, the absorption of UV light. So the more ordered it is, the more ordered the structure, it will be less. That means this particular structure will absorb less UV light at 260, whereas this structure will be slightly, there will be increase in absorption, and here it will be having more absorption. So as the more ordered the structure is, the less light is absorbed. So experiment was conducted where it was shown that the double-stranded DNA, that is this phase, okay, that absorption is 260. So at this stage, the absorption or absorbance of light was 1.00, and at this stage, the absorption was around 1.37, and at this stage it was around 1.60. So this states that free nucleotides absorb more light than the single-stranded DNA, and then single-stranded DNA absorb more light than the double-stranded DNA. Okay, so now if a DNA solution is heated slowly and the absorbance is measured at various temperature, you will get a melting a melting curve is obtained. So you get what is known as melting curve is obtained. So if you can plot a graph, or you can notice it's not a graph. So if you see, you take a DNA at the initial stage, it's a double stranded. As the temperature increases, it starts unwinding. That is, the hydrogen bonds, the bases will start breaking up, right? And slowly, at one stage, the two strands which were tightly bound will finally be individual strands. So this here you can see, before the rise the molecule is fully double stranded. Yes, then the rise began, so the base pair are broken, and the base pair are broken, and it increases with the increase in the temperature. Now critical strands get separated at a critical temperature. So the temperature at which the rise in absorbance at 260 is half complete, that is this point. This one is known as melting temperature. And melting temperature, which is designated as Tm. So you can say at 260, absorbance at 260 remains constant up to a certain temperature. It remains constant to a constant temperature. Well, this temperature um, is far above than the normally the temperature which is being encountered by the living cells in nature. And you can see that the rise in absorbance occurs at around, so if it is 70 and here it is 90, so in between it happens. So the rise in the absorbance occurs usually in the range of 76 or 78. Okay, now the maximum absorbance is about 37% high than the starting point. So this is all about denaturation and melting curve. Now I would like to talk about a term which is called as hypochromic or hyperchromic. So hypochromic or hyperchromic hyperchromicity describes a material's decreasing ability to absorb light, and hyperchromicity is a material's increasing ability to absorb light. So let me write it down here. Hypochromicity, that is this, means it decreasing ability to absorb light, and hyperchromic city is the material's increasing ability to absorb light. So what is hypochromic effect? It actually describes the decrease in the absorbance of UV light in a double-stranded DNA compared to its single-stranded counterpart. Counterpart. So this is a general introduction about what we are going to see, that is the factors that determine the structure of the DNA. So this is all about the introductory part. Now let's see each one of them one by one. So the first one is hydrogen bonding. Let me just take a page. The first one is hydrogen bonds in DNA. So the melting temperature, okay, the melting temperature increases with the percentage of G plus C. Yes, the fact is interpreted in terms of the hydrogen bonds which are present. So you know that adenine pair with thymine with two hydrogen bond, whereas guanine pair with the cytosine with three. So that this explains the statement here, right? So it requires higher temperature. So very obvious, it requires higher temperature is required to disrupt the GC pair than a T pair. So you can say that GC helps to stabilize the DNA structure more than a base pair with thymine. Right, so this is all about the hydrogen bonding is something which you know. I just pen it out the point as a point. So the in here I just want to mention something, that is urea plus formaldehyde helps to clear the hydrogen bonding in the just hydrogen bonding with DNA. It helps. Just want you to just keep that in your mind. DNA bases. So the next point is hydrophobic hydrophobic interactions. Talking about hydrophobic interaction, so there was certain experiment which was conducted, and what happened, what they did is the reagents which were used in the experiment, they were one which can enhance reagents which can enhance the interaction. I don't know what's happening with my pen. It's getting stuck. Interaction of soluble substances with water, and next is that disrupt the water shell. So they actually used reagents which actually is hydrophilic and hydrophobic. So hydrophilic, as you know, the name suggests, it is water loving, and hydrophobic is water hating or does not interact with water. So he used two reagents, that is for hydrophilic, he used, they used methanol, and For the hydrophobic, they used sodium trifluoroacetate. It actually weakens the hydrophobic interactions. So this was like they used these reagents. So when they actually added both of these hydrophobic and hydrophobic and the hydrophilic reagents, what happens is it actually melting reduce the melting temperature. So so this proves that this type of interactions are important in stabilizing the DNA structure. That is, hydrophobic interaction is like they conducted an experiment where they used methanol or sodium trifluoroacetate, okay, which is one is hydrophobic and the one is hydrophilic. And when they added these two reagents, they found out from that experiment, right, that it is going to reduce the melting temperature, then which definitely automatically proves that this type of interaction is essential in the stabilization of the DNA structure. So in double-stranded DNA, double double-stranded DNA. So if you see the structure, okay, let me just write it. You see the portions, you know that this is where the bases interact with each other, right? It's a place where the bases, and these places are usually hydrophobic, and the sugar phosphate bond backbone are usually hydrophilic. In nature, okay, this point you need to remember it. So this is the part which is going to interact with water, and our nitrogenous bases does not interact with water. So keeping this in mind, they have conducted another experiment where they actually used substances to increase the solubility of adenine. So they saw that as there is an increase in solubility, there was a decrease in the melting temperature. And that is lower of the temperature, that is the lowering of the uh, melting temperature is connected with the increase in the solubility of adenine. Okay, so remember one thing, this part, the sugar phosphate bond, it is not going to interact with anything. It does not have anything to do with the, as of now, it does not have anything to do with the stabilizing of the structure. But whereas you know that the nucleotide bases present inside are hydrophobic, so they have used substances which will actually increase the solubility of these nitrogen bases. So when they saw that as the increase in the solubility was observed, there was a lowering of the melting temperature. Okay, so this was what they concluded. So in order to just confirm that this has an effect only on the bases, okay, not on the sugar phosphate backbone, they used sodium pyrophosphate. Okay, to check it. So what this sodium pyrophosphate, it is a highly charged molecule that precisely has a solubility property which is similar to the phosphate, sugar phosphate backbone. Okay, so they just added along with this adenine plus this, they added it, and when they saw the reagents actually increase the solubility of the adenine. When the solubility of adenine was rising, there was melting temperature was lowering, and it does not have any effect on the sodium pyrophosphate. Is it clear? So just imagine, please, it's like adenine, you add a substance to a solution which contains adenine, it will make the adenine solubility property increase, right? Then what will happen simultaneously? It was noted there was a decrease in the melting temperature, right? So in order to test that, it does not have any effect on the sugar phosphate, then they took the sodium pyrophosphate, okay, which has a similar solubility property as of the sugar phosphate backbone. So what happened, it does not have any effect, the reagent did not do any harm to this, but yes, there was a further increase in the solubility of adenine, and also there was a decrease in the temperature. So this actually proves that um, hydrophobic interaction does add to the stabilization of DNA structure. Okay, now let's, in the next video, we'll talk about base stacking and the rest of the part. So thank you for watching. Thank you.